Welcome to cOmicsART, customizable omics analysis and reporting tool. Today we're going to show you how to use this application to aid in your statistical analysis. The way cOmicsART is designed is in a tabular format. Currently you only see the data selection, but if you upload your data and go through the steps, progressively more tabs will be shown. Let us start by uploading our data. The data will be uploaded with 3 CSV files. The first one is the data matrix, showing the counts. On the rows they are the entities, for example genes. On the columns they are the samples. The sample table shows you meta information on the samples, while the entity table shows you any information on the entities you have. If this was too fast and you need more information, you can always, throughout all the sessions and wherever you are right now, click on the documentation. It will bring you to our website with an extensive documentation on what to do. Another option is to click on the helper buttons, these small question marks. Wherever they are, they'll show you some kind of help for whatever you're doing right now. In this example, it shows you how to upload your data via the file input. It shows you again what it should look like, what it contains. And what is important is that the rows of the data matrix correspond to the rows of the row annotation, while the columns of the data matrix correspond to the rows of the sample table. Okay, so let's just upload our data. We choose an example data set. You can also download this. We just choose the read counts, the sample table, it's a sample sheet, and the entity annotation. When we've uploaded this, the first thing we want to do is we might want to check how is our data interpreted in this program. For this we click on Inspect Data. We need an additional click on Upload Data for visual inspection, and then it will show us how does our program interpret our data. Pitfalls are here, for example, if we choose the wrong separator, then it will only have ever one column, and you see that this will be interpreted as wrong. Does it show all the other information? And then down here there are a multitude of checks where you can check whether your data looks as it should be. Green means fine, red means you need to go into your data and change things, while orange means yeah, it's not good, but we can handle it. For example, in this case it's NA values or missing values in the data. The application will just remove those entities from the table. And then we just click on Upload New Data, and as you can see, we now see our result part of the design, which in this case is the data selection. The first thing we can do is we can add annotation to the data if we didn't do that. We did this already, so we don't need to do it, and you can choose the organism to add the annotation. And then you choose row or samples. For the sample selection, you first choose among the columns that you have in your sample table, and for example you can choose the cells, you can also choose the condition, or you choose the experiment or the sample, and you might want to select only specific samples you want to include the data. Perhaps there are strong reasons to exclude some, and you just click on them, you remove them all, and will only use these data sets. You can do the same for the condition. Here it's treated or untreated. If you have multiple conditions, you might want to only check one of them. We want to use all data. The same can be done with gene, with the row selection, and you can choose either the gene names here in ensemble genes. You might not know those gene names all by heart, so you select the symbols, and you can select which ones you want to use. You can also use the gene biotype, and which shows you functionals. So for example, in transcriptomics you sometimes only want to use the protein coding genes. So you can select protein coding and will only use all of the protein coding genes that you have within your data matrix. You can do the same for processed pseudogenes, and it will select all the processed pseudogenes. In our case, we want to use all our data, so we just start our journey by clicking on this button. Clicking on this button will directly bring you to the preprocessing, and here you can see again the design choice. On the left side we have the sidebar with all the options that you can choose, separated between things that you need to choose and then click on the button again, or things down below this black line which you can just change. For example, if you choose the DSEQ sequencing, you can choose the genes If you want more information on what kind of pre-processing we can do, you can click on the helper button or go to the documentation. You can also choose batch effects from the columns you already have in your dataset, though keep in mind, usually batch effects are only done later when realizing that there are actually batch effects. So in our case, we want to do a log10 scaling, so we need to click on this before clicking this button and then get the pre-processing. This will take a bit of time, but then when it is done, the results will be shown. In this case, a distribution matrix, some kind of statistics, show you what changed from raw data to pre-processed data. And as you can see above, all the different tabs are now shown as well, from sample correlation, PCA, significance analysis to Heatmap. To show you an example how the options below the bar work, we just click on condition, for example, and without having to re-click the get pre-processing button, in time directly the plot updates, showing you potential similarities based on the sample condition you selected. To note that now that all the tabs are shown, we can always switch between any of them, do the analysis and go to any other. We'll start with the sample correlation. Here again, by design, on the left side is the sidebar, which lets you choose all the options for the plot. In this case, above the button, things that need recomputing. You can always click on the help files if you are not quite sure which kind of correlation method, for example, in this case, you want to choose. And down below are things that can directly change the plot. If you click on get sample correlation, the sample correlation will be plotted. And you can see it is already plotting the cells, the samples by the cell names. We can add the condition to see does it plot by anything else, and see kind of clusters and patterns. Down below, where we'll go next, you can see that for each plot we'll have save plot, send only to report, or get underlying arcode. Save plot is fairly straightforward. You choose the file type, you click on save the plot, and will open up to just directly save it. What you can also do is you can get the underlying R code and data to directly, it gives you the exact R code you need to reproduce the plot, and then you can adjust it to your liking. Or you can send the plot towards a report, which will send the plot along with any kind of options you chose, like the Pearson correlation that you colored the samples by cell and condition, along with nodes that you can take manually towards an HTML report to save it for later. This enables you, when you download the report as an HTML at the end of your statistical analysis, to reproduce any kind of findings you had within the application. So it shows you what kind of options did you choose, what was the pre-processing, how did you produce it. And this send only to report, get underlying R code and save plot will always show up below any plot. Let's continue with the principal component analysis. Here again, we can select data, or we just get the principal components for now, which will open the principal component plot. Now, if we want to select data, this is only advised for really looking at variance explained within specific data points, but otherwise just get all the data, get the principal components as you know it. And then down below, we can select what do we want to color them after, what kind of principal components do we want to see, what do we want to show at the end, tooltip if we go over it to hover and see what are perhaps outliers. Again, we have the send only to report, get underlying R code and data, and save plot as everywhere. We can take notes and then send it to the report if we want to. Or we can say, well, let's directly color it by the condition, because if we had treated and untreated, do we see a clustering here? We can see more of a clustering, but it's not the first principal component, but the second one. So we can just reselect the principal components that we want to show on which axis. We can also switch to the loading plot, which shows us for each principal component that we currently have selected on the x-axis. The loadings, again, we can choose between the sample annotation, meaning we can choose the gene biotype, we can use the gene symbols and everything else. In general, on the left side, again, for the design, options to choose, separated by the button, with above the button needs recomputing, below the button directly updates. And on the right side there are the results. For example, in this case, different kind of PCA plots, like the PCA plot, the loadings, the loading matrix, or the street plot. And below the plot sometimes are additional options we can choose to change the plot. And then lastly, there's always the send only to report, get underlying R code and data, and save the plot. In the significance analysis, we again start by choosing our options in the sidebar, choosing the level of groups we want to compare, like the condition, treated or untreated, or we choose the cells. We can choose more than just one comparison, and by clicking on the Get Significance Analysis later, it will directly do all the comparisons we need. We can choose the test method, the significance level, and the test correction method, and then just click on it. And it directly runs all 3 comparisons we have selected. Up above where the analysis is running, when it's done, we can see, okay, it shows us we didn't find anything that was significant. But in case we're in an exploratory case, we can also have a look at the raw p-values, though we have to treat this with caution. We can see here, for each comparison, we have a table or the volcano plot. In the table, we can sort by whatever means we want to. For example, the p-adjusted values, we see all of them are 1. But some p-values are significant. We can download those tables as CSV files, as Excel files, or we can copy them to the clipboard, or we can have a look at the volcano plots. In the volcano plots, it shows us the volcano plots with corrected p-values and uncorrected p-values. Again, we can interact with the plot. We can choose different thresholds to show what are we considering as something noteworthy, and as in every plot, we can send it to the report, get the underlying R code, or save the plot. In the result visualization, which is currently empty, it is empty because we have nothing significant. But if we only look at raw p-values, we can get a comparison. And what it does is it shows us for all the 3 comparisons, or any others we did, shows us intersections of those significant genes in our case. And then we can compare which of the genes, for example, we have 62 genes that are significant in all 3 comparisons. Does that tell us anything? What are those 63 genes, or what are the 105 genes in the first and second comparison? We can highlight this, we can download the report, and again, for the complete plot, send it to the report, get underlying R code and data. We continue if we want to have a look at a specific gene. For example, we didn't find it. We can select between raw or pre-processed data and the single gene visualization, and it draws us box plots or comparisons, because if we only have 2 data points, it doesn't draw box plots. We can choose what do we group these things after, and so if we go to treated and untreated, now we see box plot and we see the comparison. Directly does the statistical test on this, and we can also see how did preprocessing change our data or change anything. Again, we can send the plot to the report. Another integral part of exploratory analysis is the enrichment analysis. In our case, we have the option between gene set enrichment or overrepresentation analysis. In the case of the gene set enrichment analysis, you'll be asked what to sort your genes after. You can choose between log fold change or absolute log fold changes. Then again, you have to choose by what do you want to compute your log fold changes. And then you can choose your gene sets that you want to compare to. We have a wide variety of gene sets. The most standard ones known are KEC, hallmarks, Go, and Reactome. And by clicking on the question mark again, you can get any kind of information, what the gene sets are, what do they contain. You also choose the test correction. Then for each gene set you did the enrichment for, you'll be shown a tab in which you can see the enrichment, or you can select the table. Again, for the tables, you can download the tables, and for the plots, you have send only to a report, get the underlying R code to reproduce this in R directly, or save the plot as it is. The last analysis in cOmicsART will be the heatmap, displaying the count matrix in a clustered fashion, where we now can select what kind of rows do we, for example, want to plot. We only want to plot a few entities, so we select the row annotation, select the entities we want to plot, well, the first 8, let's say, and then just click on the button Get Heatmap. again, get Heatmap just as it was. Get gene set enrichment analysis, get PCA, get significance analysis. If we don't see any differences, we can also do a row-wise scaling to make differences more clear. We can select what do we annotate the samples or the rows after, and again can send everything to the report or save it as a plot. As you can see, all the design choices are the same. Things that are above the button on the sidebar will need a recomputation, things that are below don't need one but update the plot directly. In the results we always see the plot or the table or multiple plots. We can download them and send them to the report. When we are done with our analysis, we can download the report as an HTML, which will create an HTML file which you can directly download and then open it. When you open it, you will see a report on everything you did and every choices you made, enabling you to recreate everything you did in the application again, making every result you did in cOmicsART reproducible. With this, I want to thank you for your attention and I hope you enjoyed the application.